So onward to alleles. Before we get there, something new in the news that's directly relevant to what we talked about last class, and we have a couple times sickle cell anemia. A new article came out yesterday, so you can read this if you want to. It's on npr.org. If you really want to type in the whole URL, go for it. But one of the first humans who has undergone treatment for sickle cell anemia using gene-edited cells. So in this case, they used CRISPR, which some of you may have heard about in the news. We may talk about a little bit later in the term. And they extracted some of her own cells, edited her hemoglobin gene to correct the mutation that causes sickle cell disease, and then put the cells back into her. And they're hoping that those bone marrow cells will take hold in her bone marrow and start producing red blood cells that have the wild type or the normal version of the hemoglobin gene in them. So she's been doing this treatment for months apparently. She just home, and they're going to find out if this actually works. Brave new world we live in. Gene editing, and we'll talk a lot more about that later in the semester as well, especially when we get to the genetically modified organisms part of the course. Is it right to do? Is it safe to do? That is it morally and ethically right and safe to do? And other things. So if you would, for a minute, please have a go at this. We've got 5 different things to do to start out, and then we'll talk about this. Circle some things, draw some rectangles around some things. We're going to review haplotypes and genotypes after you've had a minute or so to work on this. Got 2 chromosomes, 13, one paternally inherited, one maternally inherited. Let me number these. Let's start with number 2. Are there any heterozygous genotypes up here? And if so, which number in the nucleotide alignment are they?
3.
3 and 9.
Okay, so let's see. So question 2, draw a rectangle around a heterozygous genotype. It's either 3 or 9. Right, heterozygous, different nucleotide on the homologous chromosomes in the same position. Everything else up there is homozygous. They're all the same. So if for question 1, if you circled anything other than position 3 or position 9, that's good. So hey, already done with 1, 2, and 3. Excellent. So then the haplotypes. What's the definition of a haplotype again? Bits of DNA that come together in the same haploid cell, which means they came together in the same gamete, which really means they're on the same chromosome. Usually haplotype means they're either different chromosomes that came in the sperm or the egg together at fertilization. More frequently, we're talking specifically about the DNA that came together on one piece of DNA. So we know that that whole chromosome, chromosome 13, if it's paternal, if this is the paternally inherited version that came from the sperm at fertilization, that this is a haplotype. What's circled in red up here? This is the paternal haplotype. It's the DNA you got from the dad at fertilization. Geneticists would write this, well, there are 10 nucleotides up there. How many of them are heterozygous? 2 of the 10. Those are the only ones that are different between mom and dad. So geneticists, when they write a haplotype, don't usually write in all of the homozygous genotypes because they're not different between mom and dad. What we're trying to do by talking about haplotypes is figure out what DNA got inherited from which parent. So we only need to focus on the differences between the parents. So a haplotype, the paternal haplotype in this case is CG. So the 2 nucleotides at the heterozygous sites. So what's the maternal haplotype in the same format?
AT.
Right, A and T. Now if you wanted to write out the whole thing, you could. You would just be copying this whole chromosome, TTCA, is the paternal haplotype. But it's most important to focus on the differences. This is different than what we saw last class a little bit. Because last class we saw, when I was defining haplotypes, we also talked about DNA on a different chromosome. So let's add that in just quickly. Let's say chromosome 2 paternal, there's an ATG at some spot in the chromosome. And chromosome 2 from mom, there's a TTG. Right, so there's another heterozygous position here, the first position on chromosome 2. So if we wanted to add that to these haplotypes, what would we have to add in question 4, right? The paternal haplotype. It's CG on chromosome 13, and then something on chromosome 2. So what do we want to put in? A, because all the chromosomes from dad are a haplotype. They all came in his dad's haploid cell, the sperm. There's just one other thing we have to add there. What's the question? What are you concerned about? How can I help? We're dealing with 2 chromosomes. There's a particular type of nomenclature we have to use to separate when we're talking about what's on one chromosome versus another chromosome. This is the semicolon. If we really wanted to, we could specify which of those chromosomes were which with numbers. We don't have to do that. So that's the paternal haplotype, CG semicolon A, and the maternal haplotype is AT semicolon T. Sure. Genotype for genetic nomenclature, using genetic nomenclature for question 1.
Yes, question 1.
Okay. So the genetic nomenclature, homozygote, this is now haplotype. We use the semicolon to separate different chromosomes, that is different homologs, different numbers of chromosomes, chromosome 13 and chromosome 2 in this case. When we're talking about the 2 haplotypes at the same spot on the same chromosome, another word for a genotype, 2 haplotypes like this. There's a C here, at position 3 there's an A. Right. C is a haplotype from dad, A is a haplotype from mom. When we look at them both on the same homologous chromosome, the 2 versions of chromosome 13, we use a slash. So that in genetic nomenclature would be C/A. Except I should have put that on the line for the heterozygous genotype. C/A. So that's a genotype. Thank you for making me stick to what I wanted to do and fill in all the answers. So then a homozygous genotype will be what? Let's say position 6. What's the genotype at position 6? A/A. So the other heterozygous genotype, just for fun, was G/T on chromosome 13. Any other questions? Yeah.
Sorry, I'm just
That's okay.
confused about the A and the T with the semicolon. Like, where did the A and T come from again?
Okay, thank you for asking. So this last bit, the maternal haplotype, the A and the T are on the same chromosome 13 from mom, so they're written just next to each other with no punctuation because there's no difference in which chromosome they're on. So that A and T are that chromosome 13 haplotype. The semicolon separates different homologous chromosomes, different numbered chromosomes. So 13 on the left, then that T came from chromosome 2. Also the maternal version of that chromosome. Thank you for asking me to restate that in a different way.
Before we move on, could you also do the genotype using genetic nomenclature for number 1?
Now we can get back to last class's Socrative, which we never really went through the answers for because we hadn't really gotten to the part where you were actually prepared to answer these questions. Maybe you're still not, but let's go back to question 2. Are A, B, and C all genotypes as written here? Are they in genotype nomenclature or haplotype nomenclature? Genotype, because there's a slash. So that's good. So the question is, the genotype is the description of 2 different homologous chromosomes, which are separated by the slash. So you've got one homologous haplotype, CT, together on the left, and the other homologous haplotype, GA, on the right. So what's the genotype? What's a genotype from this nomenclature, CT/GA?
CG.
CG. Because if you write out CT as in the sequence of a chromosome, like we just looked at in the previous slide, and you write out GA, there is a genotype C/G and another genotype T/A. 2 different parts of the same chromosome. So in other words, to reiterate, this CT/GA combines haplotypes and genotypes in the same nomenclature. CT is a haplotype. That's why when I draw it out, I write them both on the same chromosome. I don't know if there are nucleotides between them or not. That's not important, because we only care about the nucleotides that are heterozygous when you compare the 2 chromosomes. So this means there's a C and a T on the same physical part of a chromosome, same physical molecule, same chromosome, and a G and an A in the same positions as the C and the T, but on the other homolog. So haplotype, one side or the other of the slash. Think about that for half a minute, and then I'm going to take a class poll. So I'm going to expect everybody to either say something or raise a limb. Your choice. Okay, time's up. Is CG, if you agree that CG is a haplotype, raise your hand or yell something. CG. No? What about CT? Okay. True. What about GA? Yes. And TG? TG is a...
Oh, yeah, it's genotype.
It's not, because if you drew out those haplotypes, the C lines up with the G and the T with the A. So C/G would be a genotype, or T/A would be a genotype. But there are no slashes up there, so there are no genotypes.
It's a trick.
It's very much a trick. But there were haplotypes up there, so that's fine. But when I ask about genotypes, yeah, then it's all a trick. Okay, genotypes and haplotypes. I think I used this analogy last class. I meant to. I'm pretty sure I did. I'd go back and look at the video recording, I suppose. Each of us is diploid, so we all have 2 alleles of every gene. So in this case, the garage is our genome, and the car is an analogy for a gene. So you can park 2 different cars, 2 different versions of a gene in your garage, unless you have a 3-car garage. But that's not diploid, that would be triploid, so we're not going to count that. The point that I forgot to make the last time, to extend this fantastic analogy even further, is that even though each of us can only have 2 alleles of a gene, because each of us only has a 2-car garage, and we can only park 2 cars in that garage, that doesn't mean that there are only 2 alleles of a gene. That is, there's not just 2 versions of a gene in all people. It just means each of us can only have 2 versions at a time. So what is the maximum number of versions of a gene or a chromosome that could exist?
Infinite.
I've got to vote for infinite. It's probably pretty close to infinite. There's a finite number of nucleotides, but it's huge. There are billions of nucleotides. So yes, there are an unlimited number of possible alleles. If we talk about any gene or any chromosome, there could be 30 versions of a chromosome. There could be 300,000 different versions of a chromosome. Those would all be different alleles. They'd all have different haplotypes. And this is something that human geneticists are pretty interested in, and other geneticists of other species too, is how many alleles are there? Why would there be so many alleles? Are some better than others? Are some, and this is where we're heading, are some versions of genes more frequent than others? That is, are some more common and some more rare? So back to our case study, allele frequencies for 15 autosomal STR loci. So STR we talked about last class means microsatellite usually, some small number of nucleotides repeated n times in a row. So allele frequencies for 15 different ones of these on different chromosomes on US Caucasian, African American, and Hispanic populations. And I showed this chart briefly last time. 13 core, these are typically genotyped STRs or microsatellites when forensic scientists are analyzing DNA samples and want to know the genetic composition of the person that left the sample. They'll usually focus on some or all of these 13 specific spots on different chromosomes where geneticists know that there are STRs or microsatellites. And the lengths of those satellites, the number of repeats, differ between different humans. So they make a really good sort of fingerprint. Presumably, every one of us, or almost every one of us, has a unique number of repeats at these different microsatellites. So altogether, they should, in theory, come up with a unique fingerprint identity for each person, or pretty close to unique. So for this, just for today, we're going to focus on the TH1 allele, which is on chromosome 11, and the TPOX. Those are just two names for the different loci, the different microsatellites, TH01 and TPOX. They're on two different chromosomes, 2 and 11. And this publication that you might have looked at, maybe you didn't, that's okay, lists for every one of those 11 markers, like TH1, how frequent each allele is. So allele, different version. Here we mean, when we talk about allele, that means microsatellite or STR length. Number of repeats. So allele 8 means the repeat unit, whatever the repeat unit is, has 8 copies. And so these geneticists basically collected DNA from a bunch of people of different ethnicities in the US, Caucasians, African Americans, and Hispanic, and they genotyped them. They found out how many repeats were on all the chromosomes in a large population of individuals. So they found, for example, that TH1, you find 8 repeats in the sample of whatever group it was they were looking at about 10% of the time, a frequency of 0.096, about 10%. So visually what this means is that if you looked in a group of 20 people, these are humans, we're diploid. So if you look at 20 people, how many alleles are you looking at? 20 people, diploid.
40.
40 total alleles. So let's see, equals 40 alleles. So if the allele frequency is 10%, how many TH1 allele 8s should we see in the whole group? This 8, the allele 8 is 10% of the total number of alleles. So if we've got 40 alleles, what's 10% of that? 4. It means if you look at 40 people, you should see 4 allele 8s in any combination in any of the people. So sometimes you'll find these gray individuals are the ones that have allele 8. Sometimes you'll find heterozygotes where one of the 2 alleles is 8 and the other one's something else. Sometimes you'll see a homozygote where they have both of their alleles are 8. But in total, in that whole population, 20 people, each of them has 2 alleles. That's 40 alleles. If the frequency is 10%, 40 alleles times 10% equals 4. So that's the number of alleles we should expect to see. If we picked a random sample of people, we should see every 10th time we look at somebody's allele, we should see allele 8. And it's exactly the same story on the right side. I just picked a different allele, but it's the same drawing. It happens to be an allele that's also at about 10% frequency. So in the same group of 40 people, you would still expect to find 4 versions of allele 8 or allele 9 of TPOX. But what's different between the left side and the right side? Subtle. It's part of the drawing. Who's got these alleles? Same people or different people?
Different people.
They're different people. So the alleles are randomly distributed in the population. Just because somebody has allele 8 at TH1, like that person, doesn't mean that they have any particular version of another allele of another gene. They're separate. And that's because these are on different chromosomes. So they segregate, they are inherited randomly at fertilization. Let's see. So CSF1P0, there on the left side. This is one of the tables from this paper. US Hispanic allele frequencies for 15 STRs. So here's CSF1PO. And that one is located here on the distal end of chromosome 5. How many different alleles are there? So that's what each row is, a different allele. Right? New numbers indicate the number of repeats of each of this microsatellite. So 7 repeats, 9 repeats, 10 repeats. But don't worry so much about the decimals, because it's hard to have a decimal number of repeats. That's a subtle thing that we don't need to worry about at this point. If you're curious, ask me after class. So what's the most common allele in the US Hispanic population at that part of chromosome 5, just focusing on this column on the left side? What's the most likely allele that you would find in a US Hispanic individual?
12?
It's whatever the biggest number is, so yeah, 12.357. So fully a third of people that were identified as US Hispanic individuals had that number of copies of that part of their chromosome 5. What's the least frequent? Whatever the smallest number is.
It looks like.
15 or 10.3, we'll just say it's 15. 0.004. So 4 out of 1,000 people, this is a really rare allele. Only 4 out of 1,000 people have that number of repeats, 15 repeats at this microsatellite. So 12 copies, 12 repeats is most common, 15 is rare. So the columns are the different microsatellites, different regions of different chromosomes, and each row is the shows the frequency of each number of repeats at these different sites. So 15 was the most rare, 12 looks like the most common. So let's apply a little bit of this information. At what frequency would you find individuals with CSF1P0 allele 9, circled in red, and TPOX6, circled in blue? Take a minute if you like, figure out how would you do that calculation, and if you've got the time, do the math. what is the algebra we should set up here? We're looking at a combination of frequencies. So if you looked in 1,000 individuals, keep working on your own if you want to. I'm just going to talk a minute more. If you looked in 1,000 individuals, how many CSF1P0 allele 9s would you see? Frequency of 2%. Move the decimal place 3 points, 1, 2, 3, you'd see about 21 individuals in 1,000 would have that relatively rare number of repeats at that locus. What about looking at in 1,000 people and finding allele 6 at TPOX? Right, 4 out of 1,000 individuals. I'm just trying to make this relevant, both. So they're both really rare. One's rarer than the other. The combination of the two, though, what's the probability that in that group of 1,000 people, somebody has both? It's the same statistical analysis we've done with everything else we've done in class so far. Independent probabilities multiplied together. So what's 21 over 1,000 times 4 1,000ths, or 0.021 times 0.004. Multiply those two together. That gives you the frequency of both. How many people should have the one and the other at the same time? It's a really small number. It's 0. Did anybody do it? 0.00084. Okay, that sounds about right. So 0.000084 frequency. If you take the inverse of that, that is 1 over that really small number, you get a big number. This is about 1 in every 12,500 people will have that combination just of those two alleles. It's even rarer, which shouldn't be surprising, I don't think. Hopefully that's intuitive. If you've got one rare thing and another rare thing, the chance of both of the rare things happening at the same time or being in the same person is even rarer. So how do forensic scientists use this information? Say you're at a crime scene. So you're a forensic analyst and you're at a crime scene and you get a DNA sample and you genotype it and you discover that the individual that left the sample had those two alleles. This DNA sample has 9 repeats at CSF1P0 and 6 repeats at TPOX. And you've got 3 suspects in the case, and you collect samples of DNA from each of them. And one of those 3 people is a match. Did that one person certainly commit the crime, or at least leave that DNA sample?
Yes. They did the murder.
They did the murder. This is an unfortunate part about criminology, is that there's no sure thing. What's the probability that any one person picked at random from the population has that particular DNA fingerprint? You'd have to be really unlucky to be, wandering past the police station when they grab you and say, "Here, give me a DNA sample," and it matched, because the frequency of this particular combination is 1 in 12,500 people. So if you're on the jury and the prosecutors come up and say, "We got this DNA from the crime scene, it matches one of our 3 suspects," probability is that that DNA fingerprint, this combination of alleles, is so rare, it's pretty likely that it came from that one person. Are you sure that it came from that person?
Not unless we sequence all of it.
Fair enough. You're only sure that that person did it if you sequence everybody else, and rule out everybody else that has that haplotype, that combination of alleles, as possibly having. You have to get the alibis from the other however many people, 1 in 12,500 of 3 billion people on the planet. Yeah, right? You'd have to do a lot of police work to to get alibis for all those people that showed that they couldn't possibly have been there at the time that the DNA sample was left. Yeah.
Yeah, sorry, just like regarding how we put it over 1,000, wouldn't we use the 140 if that's the population size for n equals 140 up into like, so like. when
Say
we
that
put-
again?
When we put like the frequencies over 1,000 individuals, right?
Oh, these, yep.
So wouldn't we use though that 140 up on the n equals 140, the like population size? Like. if
Oh,
it's-
this, sorry.
Yeah.
Sample size was 140.
Yeah.
So wouldn't we put it the frequencies over 140?
Thank you for spotting that. I think the answer is no, but let me explain the n equals 140. Well, I'm glad you asked the question.
Sorry.
No, that's totally fine. Hopefully that's what you're here for, is to ask questions. So the 21 out of 1,000 is accurate, but you're absolutely right. What they did was they collected DNA from n equals 140. 140 different people that either the scientists said were US Hispanic, or those 140 people self-identified as US Hispanic, one or the other. And they found that of those 140, actually I don't know in this case if n is the number of people or the number of alleles. So it could be 70 people and 140 alleles, which is more likely. So we're just talking about this n equals 140. I'd have to look back at the paper and find out if it was 70 people or not. So you're absolutely right. This 0.021%, that's frequency from those 140 people. I'm just extrapolating that to make the math easy, multiplying that number. So yes, these data came from this population of 140 people. But you can also amplify that 140 to 1,000 just to make the point that it's a really rare allele.
But if we used it, we would basically still get the same.
Exactly. So we could reverse calculate if how many alleles were there in that group of 140 individuals or alleles if the frequency is 0.021. So what's 2.1% of 140? That's the number of times they would have seen that allele in that small group of people.
Okay.
Yep. And this brings up a really important point. This is a model. That is, this is some data that some scientists collected and published. To what extent are these numbers that are in this table fact? And are there any limitations to this model? So I've explained one way you can use this model to make predictions, for example, in the setting of forensics. Is there anything misleading, wrong, unuseful about this data set?
So the limitations could be that this was taken from a small group of people from like, if everyone lives in a small town, then everyone's gonna be like, man, that's gonna be creepy if I said semi-related. But like,
Well, we're all related, so that's
yeah,
we're all
they would
humans.
be more close in genetic information. If they took just like a random like smattering of people from like, oh, okay, one from New York, one from California, one from Oklahoma, one from, then it'd be a lot more diverse, a lot more representative of the U.S. population. But
So-
this is not like for everyone in the whole world.
Right. So one limitation, how representative of people in general is this table?
It's U.S. Hispanic people.
So there's one caveat. You can't use this necessarily for every ethnic group because different ethnic groups will have potentially different patterns of allele frequencies at these same markers.
And can't you just use it for those 70? Because you don't know if every single person's going to be better.
So as was mentioned earlier, they didn't sequence everybody. So this is a small sample size that these authors are hoping, they're predicting is representative of the whole population of however many millions of US Hispanic people. So how accurate are these if you do the sorts of calculations we did? How predictive are they? How useful would these be, for example, in the legal system? And this is why people get paid boatloads of money. When I was a graduate student, I worked in a lab with a research fellow, someone who had a PhD but wasn't a faculty member, who was an expert witness in DNA fingerprinting cases because he was an expert in doing this sort of math and knowing the genetic differences of allele frequencies. He got paid like tens of thousands of dollars they'd like fly in places to be an expert witness in testimonies. So if you're looking for the big bucks and you like genetics, consider becoming an expert in DNA fingerprinting because you can make some pretty sweet money. Yeah.
I'm also noticing that there's a lot of blanks or where they didn't find anything, like FGA, they found nothing on any of the alleles, and I'm like, well, that's not helpful.
I'm glad you pointed that out as well because this raises another point, which I forgot, but is important. And then I think we probably ought to move on to talking about the test for a couple minutes. If these are frequencies, then each of these columns should sum up to a particular number.
1.
And it turns out for CSF1P0, those numbers do actually sum to 1, but there are gaps in this data set. That's not necessarily bad, it just means there's missing data. And that could be due to several reasons. But that's another potential caveat, is you always have to be skeptical about data. If you find one of these columns that doesn't total 1, what happened? Where's the missing data? All right, so we're cruising through the vocabulary and tasks. At this point, we've only touched on the first task, genotypes and haplotypes. We've gone through much of the vocabulary up until the point where we start talking about PCR, denature, anneal, extend, primers, DNTPs. We're going to start talking about that on Monday. And we've started talking about the genetics class trailer question, how accurate is DNA evidence? Okay. I've got good news. Not necessarily for every one of you, but for most of you. I just want to put everybody's brains in perspective if I can, at least help put your brains in perspective about the exam. So first, mechanical stuff. I will be returning all the exams on Google Classroom. I'll press a button, it'll send back to you. And at the top of the PDF or first image file, if you submitted screenshots, I've put a comment I hope you will be able to read. And it's going to be at the top of the first page, and it will be in that format. And this is just how I scored. It's the number of points you got on the first question, second question, third question, and so on. So that's how you find out what your score was on each of the questions in the exam. If you can't see it, for some reason, let me know. I'll email you the numbers. The totals are going to be posted on Canvas some point later today or tonight or tomorrow when I have the time. So you'll see the total score on Canvas, regardless of whether or not you can see the individual scores of the questions. And then I'll also post a video key. So for you to get feedback on the exam, what I suggest is you look at the points that you earned, figure out which questions you missed points on, look at the key, and then ask me questions later about, if you have questions about why I looked at the key, I saw your answer to the question, I don't understand why I missed points, I'm always happy to have those conversations. But I make a video key specifically so that I can talk through the answer to the question as I solve it, so it should be, hopefully, helpful to see how I intended the questions to be solved. So here's the putting everything in perspective. I've already talked to a few of you, and I know a lot of you are concerned about the exam. It was a difficult exam. Most of you haven't seen exams in this style before, not only just the technology aspect, but also the way it was written, the types of questions that were on it. So I want to remind everybody that the grading scale is not the same grading scale that you're also used to seeing in the class. That 0 to 20 points shows that you have foundational knowledge. If you earned any points, that means you know something about genetics. But that's the bottom 20% of points. It's not like 50 to 60, up to 50% of points is an F. It's just the bottom 2 points on the test. Right? Test 1 was 10% of the total grade. It was worth a total of 10 points. So if you look at the syllabus and you just want to figure out how many points are available in class, it's 100. 20% or 20 points based on attendance, 35% on quizzes and assignments. So 55% of the grade, if you just come to class, do the Socrative activities, do the activities like the Canvas discussion board posts, that's going to total 55%, which gets you into the C range. Right? Basically, the exams are for me to figure out and for you to show me where you fall in the CBA range. And the first test, as we've discussed before, is the lowest percent, except for the final. So in other words, if you come to class all the time, you do all the activities, you're pretty likely going to get a C. The question then on the exams is, where do you fall from C, B, or A. So of those 10 points on test 1, then basically the first 2 points were showing me foundational knowledge. The next 2 points out of 10 were, are you developing as a geneticist, or are you C, 5 or 6 points, competent as a geneticist, and so forth, in my terminology, the way I define A, B, C, D, and F. In other words, 5 out of 10 is not as bad as it normally sounds, because that's C, which is probably bad to many of you, but it's not an F. It's a C. So here's the score distribution. So there was a perfect score. Nobody missed everything. Everybody fell somewhere in between. This is pretty typical curve for the class. If you are in the 5, 4, 3, 2, 1, I would love it if you'd come to office hours and ask me questions about how I can help. I want to. You'll find if you're one of those people later. Here's a little bit of the detail. If you want to zone out for a second, that's okay, but I think it might help you if you understand how I write questions and how I assign points to questions. Maybe it'll help. So after I'm a geneticist. I love data analysis. After the exam, I collect all this data, and I do all sorts of data analyses to try to figure out, did I suck at writing one of the questions? Is there something that I need to go back to class and help everybody learn better because I wrote a question and everybody missed it? So I've arranged these questions, the 8 questions on the exam, by the class average. That is 93% of the available points, all 50 of you say, not 0.93, 93% of 1 point was the average. In other words, most people got that question right, only a few people got a 0. So there were 2 questions that were 93% class average, and then it goes down as low as 27. So like a quarter of you got the 1 point that question was worth. The reason I'm telling you this is because this is how I write questions. Because it's a 20, 40, 60, 80 grading split, I write 2 questions specifically that I intend maybe only 20% of the class to get. Those are questions, in this case, questions 4 and 2, each worth a point. One of them, half the class got it, the other half didn't. One, about a quarter of the class figured out the answer to that question. That's why this test seems so hard, because I have to write a number of questions that I imagine that only a few of you will actually get. So don't necessarily stress out if you see a question and you don't know how to answer it. That's somewhat intentional. These are questions, bless you. These are questions, if you go back and look at the exam, questions 2 and 4, where I'm asking interpretation questions and invention questions, the most difficult types of questions. Create something. The labeling types of questions, label, multiple choice, true and false, the things that I write specifically because I think most of you are going to get it, are down here in the foundational. Can you define something? Can you identify something? Those sorts of actions on those questions, questions 8 and 7, where most of the class did get those questions right. So this wound up working almost as I expected in terms of the easier questions that I thought were easier, most of the class got, and then it falls off as the difficulty of the types of questions increased. The one problem, and this is the good news to end with, is I discovered that question 3, which I intended to be sort of middle of the road difficulty, it was obvious from responses that I was shit at writing that question. And that's why it says down here at the bottom, my disclaimer. My wording on this question was inadvertently ambiguous. That's what I meant earlier when I used the swear word. I didn't mean to be ambiguous, but I wrote a question that a lot of you answered in a lot of different ways. That was this question. 2-point question. Part of it was supposed to be easy, so one of those points was more like a D level, like you understand what you're doing, you've been to class, you've been paying attention. That was the consensus sequence. Most of you did that, most of you got that point. Not all of you, but most of you did. The one change you could make to better optimize the gapped alignment is the part where that was way too ambiguous. I know a lot of you didn't get that second point. So the good news is, if you got 1 out of 2 points from having put the consensus sequence on there, I'm going to assign the second point because That was my bad. I just wrote a question that nobody answered the way I thought it was going to. So if you got 1 out of 2, you get 2 out of 2. If you got 2 out of 2, congratulations, that was fantastic. There's no extra credit. You don't get 3 out of 2. Yeah, but there were only a couple of you that got, there were like 5 or 6 of you, I think, that got full credit on this. So watch the video. You'll see the video key. You'll see what I was intending, but don't penalize yourself for your answer on that. If you got 0 on this question, then you don't get an extra point. Because most of the people that got 0 either didn't provide the consensus sequence in the first place, or you skipped the question entirely. So you had to show some effort on this question to qualify for getting extra point. So, oh, am I out of time? We'll skip this stuff. I'll come back to it next time because I do want to tell you about your responses when I asked, what will you do differently next time to study? What can I do better to help you on the next test? I want to tell you about that Monday then. For Monday, this. So we're still on the same topic. Just please make sure you've watched the PCR and agarose gel electrophoresis videos and have tried all 5 of those exercises in the manual. I'll see you on Monday.